I'm the Associate Dean for Research in the School of Medicine, and this is the inaugural of our research brainstorming sessions, which the goals are up here briefly for everybody. Virtual meetings, everyone across our campuses is invited to attend. The goals really are to encourage development of research projects, connect collaborators interdisciplinary and across our campus, engage learners in research, and eventually for at least some of this work to help facilitate competitive proposals for funding. We have a number of speakers really on the basic science side that have volunteered to give future presentations. I think Dr. Tu will be the next one speaking, but we really do need some clinical presenters. So, if you have a project or if you know of a nice clinical project in development, that would be great. The intent is to have 2 projects presented on a given day. As we do today, we have Dr. Attila Roka from the Department of Medicine presenting non-pharmacological, let's see if I can't quite read it, rate control of atrial fibrillation using perihis pacing, about a 15-minute presentation, and then time for discussion and questions, followed by Dr. North from the Department of Biomedical Sciences presenting regulation of intracellular communication by BUB-R1, a role in controlling cardiac electrophysiology. So I'll ask you a couple things. One thing, please mute yourself unless you're speaking. And then also, if you would like to make sure that you receive an email announcement of the next session, please put your email into the chat and we will collect those. And I believe the next event is going to be, I think it's Friday, April 25th, I believe, or maybe the 29th. Sorry about that, but I'll send out an announcement. And then I don't want to take up any more time of our speakers. I thought Dr. Roka might go first, if that's agreeable to both our speakers. You can go ahead and share your screen. And thank you so much to our speakers for doing this, and thank you all for participating with us today.
Dr. Hassan, thank you very much for the introduction. Am I sharing the screen properly? It doesn't show up on my screen very well. Okay, very good. So this is a translational / clinical project we started a few months ago, and I really appreciate the opportunity we can present some preliminary results, and hopefully we can open up some opportunities for collaboration. So I would like to talk about non-pharmacological rate control of atrial fibrillation using parahisian pacing. So during a normal cardiac cycle, the heart gets activated from the sanoatrial node, the electrical signal gets conducted to the AV node, the His bundle, and the ventricle activates to the conduction system of the heart. So this is a very, very structured process. On the surface EKG, we see a P wave for the atrial activity, QRS, and the T waves for the ventricle activity. So everything activates in order, and usually the cardiac cycle length is very regular in sinus rhythm. Atrial fibrillation, which is the most common sustained arrhythmia encountered in clinical practice, there is very disorganized electrical activity in both atria, both atrial activating at the same time, and multiple fibrillatory waveforms are running all around both chambers. And these waveforms actually constantly bombarding the AV node and leading to a very, very irregular ventricular cycle lengths in atrial fibrillation. On the surface EKG, what we see the atrial activity as a fibrillatory waveform. We don't see distinct P waves. And of course, random waveforms hit the AV node at random intervals. And that's why I see that the RR intervals, the intervals between the R waves is completely irregular. This is what we call irregular. So this is atrial fibrillation. So the clinical significance of atrial fibrillation is already quite huge, and the number of patients living with atrial fibrillation is increasing. This is some estimated numbers of patients living with atrial fibrillation just in the United States. Right now, we are around 8 million, and we can see this is a disease that mostly affects older patients. As the population keeps aging, more and more patients will have this condition. So it is very, very important to try to find a way that we can treat this condition effectively and also alleviate the patient's symptoms. When we talk about treatment of atrial fibrillation, we are really talking about three very important goals. So rhythm control, which basically means that we would like to restore and maintain the normal sinus rhythm for everybody. It's not always possible because atrial fibrillation is related to structural heart disease in many patients, advanced atrial remodeling. We can't restore normal sinus rhythm in unfortunately, many cases. Rate control, if the patient is in atrial fibrillation, we can't just let the AV node activate the ventricle at an uncontrolled, very fast heart rate, but we usually use medications or some other techniques to slow down the AV conduction and maintain a more regular heart rate, even in atrial fibrillation. If the patient gets too tachycardic, persistent tachycardia, they can start having heart failure or cardiomyopathy related to the very fast abnormal heart rate. Or the patient gets bradycardic, they can get symptomatic from bradycardia related to, they can get presyncope, syncope, and all issues related to a very slow heart rate. So we need to control in atrial fibrillation from both extremes. And the third major goal for atrial fibrillation is stroke prevention. If the atria are fibrillating, there is a very inefficient contraction of the atrial musculature, the blood flow slows down, and one of the major complications of atrial fibrillation is the left atrial appendage can start forming thrombi inside that can embolize, and of course the most dreaded complication is ischemic cerebrovascular events or peripheral embolization. So most patients who are at risk for developing thrombi in atrial fibrillation, they are either anticoagulated or more recently we can actually try to exclude or eliminate the left atrial appendage to decrease the risk of clots. So of course, if we can just cure everybody and we can keep everybody in sinus rhythm, we wouldn't be here talking about rate control of atrial fibrillation. But unfortunately, we are still very far from complete rhythm control for everybody. Currently, atrial fibrillation ablation, it's an invasive interventional technique. It is the most effective treatment option for patients to keep them in sinus rhythm. And even if patients, if they undergo multiple ablation procedures, use medication, the chance of long-lasting successful rhythm control is unfortunately still low, and that's related to most of the comorbidities which we may not be able to control in every patient. So if we see that the patient had an ablation at 0 months and the patient clinically had persistent atrial fibrillation, which means that it did not terminate by itself before, after 5-6 years, only 60% of patients will stay in sinus rhythm after multiple ablation procedures if needed. And if the patient had long-standing persistent atrial fibrillation, basically means that the patient had atrial fibrillation for more than a year, the success rate for even multiple ablation procedures is even lower. So just a few years heading out, very few, less than half of the patients will stay in a normal sinus rhythm, even with the best treatment options available. So we can't just focus on rhythm control. If the patient falls back in atrial fibrillation, we still have to make sure that the heart rate remains controlled during the arrhythmia. So all three major issues related to the atrial fibrillation, rate control, rhythm control, and stroke prevention are very focused areas of research these days. And we can actually see just the number of publications in PubMed since 1980s. So there were some publications, usually when the 2000s, when the number of publication and research started increasing regarding atrial fibrillation management, and especially that was related to some newer techniques that were developed. So companies can kind of market their devices, do more research, and that would actually increase the research related to atrial fibrillation. So regarding rhythm control, atrial fibrillation ablation first was performed around 1998. And ever since, the number of AFib rhythm control publication increases. That's the blue dots. So we don't have that many new medications, unfortunately, for the pharmacological management of atrial fibrillation. The last new medication that was approved was dronabinol in 2009. That was unfortunately not a success either. So really the only advance we made in AFib ablation management is ablation. And now we know that risk factor modification also plays a really big role, sleep apnea screening and managing all the comorbidities. But really that the non-pharmacological rhythm control what drives the increased efficiency of rhythm control. The other advance in atrial fibrillation management is stroke prevention. There were actually major advances, like for decades, patients had to be on warfarin or coumadin as a blood thinner to prevent them from thromboembolic complication, which was a very difficult medication to take, requires close monitoring. all the dietary restriction related to the medications. The first breakthrough in this regard was the first novel new anticoagulant, which was the dabigatran. Since then, we have a couple more of those new medications. These medications are not diet restricted. They work for everyone, much easier to take, less strict monitoring. So this really helped patients. And of course, lots of clinical studies were done with this medication. You see how many publications are regarding stroke prevention. They still increase the risk of bleeding because they are anticoagulants. So the other alternative approach to stroke prevention is eliminate the left atrial appendage, either with an occluder device similar to Watchman, or more recently the amulet device, or sometimes surgical approaches. It can also be used. But really what revolutionized the research in this field is percutaneous left atrial appendage closure. And you can see this is a field that's under intense research right now, lots of publications being done. Unfortunately, you can see rate control is a little bit lagging behind. So everybody kind of assumed that, yeah, we have medications, same medications we had for 50 years for rate control, and they work, and well, that's what it is. We just use this medication to control the rate for patients in atrial fibrillation. So no new medication, no new techniques. So that's why, unfortunately, research was lagging behind this field. That's why if we can come up with something more efficient in this field, probably patients would benefit. So one of the trials that actually looked at the efficiency of rate control in atrial fibrillation was the RACE-2 trial, which compared how strictly we have to control the patients in atrial fibrillation to decrease the outcomes. So the problem with these studies, they take a couple of hundred patients and they always analyze easy to make endpoints. So they kind of go for the major clinical endpoints. You see this study looked at the primary composite outcome: death, hospitalization, stroke, embolism, major bleeding, arrhythmic events, side effects of medication, implanting of pacemaker defibrillator. Some of this really has nothing to do with how much heart rate we control, like systemic embolism, that's an anticoagulation endpoint, or major bleeding, that's an anticoagulation endpoint also. And unfortunately, they kind of dumped everything together in one single endpoint. And one of the reasons the study really had no difference in outcomes. Lenient rate control was that they aimed for a heart rate less than 110 beats per minute. Strict rate control was when they aimed for a heart rate less than 80 beats per minute. And overall, when they did the statistics, there was really no significant difference in the outcomes of this composite endpoint. So not a very good study. And the only factor they looked for rhythm control is what was the average heart rate of the patient, which may not be efficient. As we know, the RR intervals are very irregular in atrial fibrillation. So it's not just the average heart rate that counts, but also how the RR intervals follow one each other. We see patients when they have like a dominant cycle length of the ventricular rate in atrial fibrillation with some slower RR interval, longer RR intervals between these episodes. Some patients have consecutive RR intervals all over the place, from very short ones to very long ones. And some patients fall in between, a little bit better rate control, but still very irregular. So it's really hard to say what's driving the symptom. Is it the heart rate or is it the regularity of the RR intervals? So what affects the regularity? So of course, lots of things affect the AV conduction in patients. So atrial activity, the refractory period of the atrial tissue just from the atrial fibrillation itself. Refractory period of AV node, like the heart, the conduction system, we can't just tetanize, can't just stimulate these tissues similar to a normal skeletal muscle, but these tissues have refractory period that will affect how fast these tissues can conduct and activate after the previous cycle. If the patient has a premature ventricular beat, retrograde conduction from this beat actually causes retrograde block in the AV node that can actually slow down consecutive conduction. Drug effects, that's what we use to control the heart rate with medications. Changes in sympathetic, parasympathetic tone can also affect this. And if the patient has more than one pathway inside the AV node, that can also affect it. This is a hemodynamic tracing of atrial fibrillation. We can see on the surface EKG that the RR intervals are completely irregular in this patient in atrial fibrillation. And this is a peripheral pulse tracing with a non-invasive monitor. We can see the heart is actually doing much work mechanically. There are lots of QRS complexes here, but the patient has significant pulse deficit. Not all the QRS complexes are leading to a peripheral pulse in this patient. Completely irregular RR intervals, variable pulse with and also pulse deficit. So this is not good for patients. Okay, so some studies actually try to analyze to see, besides the heart rate, what else may affect the symptoms in patients if they stay in atrial fibrillation. This was a really elegant study. They studied 38 patients with persistent atrial fibrillation, and actually they tested them on 5 different rate control medication: baseline, beta blockers, and calcium channel blockers, and they did 5 consecutive Holter monitoring for each patient. They did not just analyze the average heart rate for patient, but they also analyzed the variability of the RR intervals with these parameters: heart rate, standard deviation, the root of the mean square differences of successive RR intervals, and also the average deviation between consecutive RR intervals. Because this is higher variability, the kind of cyclic variation of the RR intervals. They also analyzed the irregularity of the RR intervals, which is not cyclic, but more random. As you can see, we go down on this axis, you can see how chaotic these signals start looking. And they used the entropy measures to characterize the irregularity of the RR intervals. So just an overall result of this study. Of course, this is just the diurnal variation of the heart rate in atrial fibrillation. When the patient is awake without any medication, they have a higher heart rate during daytime hour. At night, the heart rate goes lower. And we use any of these medication that kind of pushes the boundaries a little bit closer to the midline. Of course, all the medications we use, they will try to eliminate the extreme high heart rates in atrial fibrillation for patients. So just looking at that, we can't say there was much different between any of these medication. But what they found that actually these patient symptoms are actually correlated with some of these parameters. So what they found that the variation in the variability parameter, heart rate, SD, if the patient has more circadian variation, which is more physiologic, they had less severe symptoms. On the other hand, if the RR intervals had higher entropy, the more randomness on the consecutive RR intervals, the patients were more symptomatic. So this is very important. It's not just the average heart rate counts, but also how irregular the RR intervals compare to each other, because that affects symptoms and hemodynamics. So there is a limit how much we can do with medications because of their side effects, and they can't really affect most of these parameters. So some attempts were made for non-pharmacological rate control for atrial fibrillation. Of course, the most drastic measure is AV node ablation, when we just ablate the AV node, put in a pacemaker, and let just the pacemaker take over. Well, this lets us to control the heart rate completely. We can stop the nodal blocker medication in this patient and eliminate some of the side effects. Unfortunately, the patient will become pacemaker dependent. This is an irreversible procedure. Even if the sinus rhythm is on, there's no way patient will benefit from that much. And also pacing from the apex causes ventricular dyssynchrony. The parasympathetic stimulation, some implanted device, we can put pacemaker electrodes close to the coronary sinus ostium, stimulate the vagal ganglia around this area. When we stimulate these areas, actually the heart rate can decrease in atrial fibrillation. You can see when the stimulation comes off, heart rate decreases. This could be actually used as needed rate control for patient. The problem, like none of these technologies matured into clinical use for technology. So this is not, we can't use this in clinical practice. And one thing that actually we can use, many patients with AFib have also sinus node dysfunction. They have built-in, they have pacemaker devices. So when we actually start pacing the ventricle, the retrograde block can actually control the heart rate for some of these patients. So this is an old experimental study, and this, I think it was sheep, and they recorded the consecutive RR intervals at baseline. It was all over the place. If we start pacing this animal at 1,000 milliseconds, of course, we will eliminate all the long RR intervals because we're pacing at 60 beats per minute. And actually, we are decreasing the number of short RR intervals because we are inducing retrograde block. The faster we pace, of course, the faster the average heart rate will be, but the faster we pace, the more of these short RR intervals are also eliminated because we are inducing retrograde block. So this was also a good idea back then. The problem is also we are starting to pace the RV apex. We are causing dyssynchrony, and the patient may not have much hemodynamic benefit if we are pacing them too fast. So one novel technology that is now used in the clinical practice is his bundle pacing. So instead of putting the pacemaker in the apex, we can actually pace the conduction system directly. And what it achieves us, instead of having a wide QRS with RV apical pacing, the pace QRS is much narrower, resembles the native QRS. So technically, we can try to use this methodology to pace the his bundle, cause retrograde block, so get the benefit from the ventricular pacing for rate control without impairing, causing ventricular dyssynchrony. So this may be a more efficient way for rate control. So that's what we try to study in our clinical, like translational clinical study. So as we recognize it, not just the heart rate, but also the variability and the irregularity of the RR intervals that leads to patient symptoms. We can pace the ventricles to eliminate the slow RR intervals and also decrease the number of fast intervals, decrease the variability of RR intervals. And our idea was to do his bundle pacing instead of RV apical pacing to make sure that we don't desynchronize the ventricles. So we actually had patients who presented for atrial fibrillation ablation or his bundle pacemaker. So we studied patients who came for the procedure, and they were in atrial fibrillation. We used a non-invasive peripheral monitor to measure beat by beat hemodynamics. We did program stimulation either with the EP stimulator system or with the his bundle pacemaker lead with the pacemaker itself. We measured paraHCM pacing and RV pacing for 60 seconds each, and also baseline for each patient. So each patient had 3 different sets of parameter baseline. We had the paraHCM pacing and the RV pacing. So this is how it looked like as baseline. This was the first image I showed before, very irregular RR intervals, pulse deficit. When we do RV apical pacing, you can see the pace QRS is wide, but we made the heart rate very, very regular. Overall, the heart rate is not faster than the native because you see we eliminated these short RR intervals, and the ventricular pacing made the peripheral pulse very regular. This is how it looks like on the electroanatomical mag. This is the right atrium, his bundle. This is the right ventricle. So we actually pushed this catheter into the right ventricular apex. That's where we are pacing from. When we did the paraHCM pacing, we just put back the catheter to the his position. You can see the pace QRS is actually much narrower. You can see a little bit of variability because some of these are actually fusing with the native conducted beats during atrial fibrillation. And you can still see that the heart rate is same as during the ventricular pacing, and the pulse became very regular, no pulse deficit anymore. So it looks very effective there. So this is how it looks on the EKG. At baseline, this patient had atrial fibrillation with very rapid ventricular heart rate. When we started ventricular pacing, you can actually see we're actually pacing slower than the average RR interval in atrial fibrillation. But once we start the ventricular pacing, that causes retrograde blocks. So this patient can't conduct that fast anymore for a couple of beats. Eventually, something breaks through, so the AFib again goes fast. Then we pace again. So overall, we can see that we are kind of pacing a little bit slower than his average cycle length, but overall, it still decreases his overall heart rate. So much more regular heart rate, not much variability. So this was apical pacing, and this is another patient with atrial fibrillation, narrow QRS. With paraHCM pacing, you can see the pace QRS is actually narrower than when we were pacing the RV apex, and also regularized the RR intervals quite a bit. So all these short RR intervals, we don't see them during paraHCM pacing for this patient. What we've had, so we had 16 patients so far. On average, the age was 62 years. Most of them had preserved ejection fraction. Since we tried to pace just 50 milliseconds faster on average than the native average RR interval in atrial fibrillation, that what came out. Electrically, the heart rate did not increase significantly. Most patients were 700 milliseconds, and it was 664 for both paraHCM pacing and right ventricular pacing, non-significant difference. The QRS duration obviously increased because most patients had a normal or close to normal QRS interval at baseline. Parahis pacing increases the QRS duration a little bit because we have some septal capture, and RV pacing, obviously, it causes much more wider QRS duration. This was significant. And luckily, the hemodynamic parameters were not affected by pacing. The concern here would be even if we pace the patient, we have a better heart rate, less variability, how much hemodynamic benefit we are going to lose because of the widened QRS. So that actually turned out to be neutral. So the peripheral pulse rate did not change. That was normal. Mean arterial pressure also remained unchanged. Stroke volume variability remained unchanged, no significant difference, and the cardiac index also remained unchanged. So none of this pacing really led to hemodynamic deterioration. Of course, these patients were under anesthesia, so it's hard to say what symptomatic benefit we would have. We would have to, since we did not see any adverse hemodynamic effects, the next step would be to do the same study in ambulatory patients when they are awake, and when they have a pacemaker, and we can actually adjust their pacemaker settings to see if the patient get any symptomatic benefit from here. So the follow-up for this study would be is one of the limitations we did is we actually used a fixed pacing rate for patients based on their average RR intervals. So it may not be ideal for everybody. We see that how many variability and irregularity parameters we can count for atrial fibrillation. So probably using these parameters, we can predict what pacing rate, what pacing modality would best for rate control for each patient. So once we have more patients, we are actually planning to develop a mathematical model to see, based on the RR intervals, what would be the best pacing rate to eliminate most of the short RR intervals, at the same time still not to pace them too fast. And if the results are favorable, this is something that can be easily incorporated into current pacemakers. There are some software changes, and this would be an extra option for rate control for patients who stay in atrial fibrillation. Okay, well, thank you very much. Do you have any questions?
Thank you, Dr. Roka. I wondered if you had funding for the study, or how are you able to do it, and how big your team was?
But this is investigator initiated, so we don't have funding. We actually started doing this as an M1 research project. I work with a medical student, so the only funding we get is a medical student signed up and they get some funding for their participation, but we did not get any separate funding yet so far.
Okay. I'll open it up to questions. I suppose we are not such a huge group. We could try unmuting if there aren't, I don't know if there are already questions in the chat.
Okay.
There are no questions in the chat currently.
Okay. So go ahead and unmute if you have a question. So I don't know if you, I guess I'll ask you, since you're presenting here, we have quite a few basic scientists and we have quite a few clinicians here too. Is there, I don't know, is there expertise you think that could help you take this really nice work to the next level of thinking about, you know, expanding the study to get it to a point where you could write up some kind of proposal for funding, or you know, move it to the next stage, or if you see that kind of future for this work?
Yes,
And
I think so. Like we are clinicians here, so I would definitely would like to collaborate with somebody who has more expensive on time series analysis, and especially when we are getting to a point when we are developing a model and to see what are the best parameters to predict the best pacing rate for patients. So they would definitely could use some help there.
So is there any animal model available so they can, I mean, this is kind of like a little bit, so you already did some clinical trial test. And I was just wondering from basic science point of view, is there any animal model available so that we can manipulate it on animal first?
Mm-hmm. So yes, so this I'm not sure if the presentation is still showing up. This graph I showed, the scattergram and how much pacing affects the RR intervals. Those were actually animal models back in the 70s and 80s. Those were acute experimental animal studies. At that point, there were no pacemakers available that could use this algorithm. So this kind of like got forgotten for many, many years, because there was just no technology to use this idea in a clinical practice. Luckily now we have the technology, so if we can replicate these results or get more human data, or even work a little bit more animal data, then I think it will be much easier to implement also in patient care.
Any like mechanism, you know, why we created this kind of cause this disease, and how do you apply for, you know, your treatment? I understand that this is more like a practical treatment for the patient. So if there's also connected to the some kind of like either genetic or epigenetic, whatever the reason can cause this kind of like a disease, and then there might be a grant, you know, submission point of view, that might be the combination would be good.
Yeah, there is a genetic component in atrial fibrillation. So we used to think about AFib as mostly like a lifestyle disease related to aging, sleep apnea, CAD, hypertension, diabetes, the conventional cardiovascular risk factors. When I was an EP fellow, actually, Patrick Elinor at Mass General Hospital, he was the one who studied the genetic correlation of atrial fibrillation with like the genetic, and he did like a very big study about like I don't really know the details, but he did like many thousand genes, and he used statistical methods to see what genes affect the risk of atrial fibrillation developing in patients independent of conventional cardiac risk factors. So this is being studied. Well, we did not study it in our study, but that's also something that being studied right now. Can we determine the genetic risk for atrial fibrillation for patients and incorporate then this information when we are treating them?
so you said expertise in time series, and I'm not sure if that would mean a mathematician, a statistician, a computer scientist to do programming of some algorithm. Like exactly what kind of expertise would you be looking for?
So I think a mathematician would be great. Like computer science programming, it's also very helpful because some of this can be simulated. Some of the recent advances in AV conduction atrial fibrillation were actually made in silico with computer simulations. the way I kind of saw those models, some of them were quite rudimentary. So I think there is still a lot we don't really know about AV conduction. So we can work on simulation, simulating AV conduction in AFib, and if we have access to animal experience, that would be also something that we can try. When I was back doing my PhD, one of my projects was to study individual patterns in AV conduction in AFib, and we used the dogs at that point. And it was a very simple experiment set up, and we had some results. But that's something that, especially if we try to optimize an algorithm for rate control, I think before we go clinical, it would be nice if we have the way to study this in an experimental setting.
So as far as the computer science and the mathematical expertise, I think Stephen Fernandez, assistant professor in the Department of Computer Science, Design and Journalism, I think it's Fernandes, I think is his last name. You know, he's very interested and a very active collaborator, especially on clinical studies. I think he might be interested. In terms of a larger project with animal studies, etc., I think this is a really good introduction to a lot of people. Maybe our next speaker who does basic science research with a cardiac bent might have ideas, or you may see possibilities there too. Any other questions, comments, suggestions?
I have a question. So is the belief based on this that this would lower, you know, in terms of you've clearly assessed safety, or at least started, this is almost like akin to kind of a phase 1 sort of trial if you were looking at a pharmacological sort of intervention. So you're really looking for like a phase 2, you know, sort of perspective here to get some efficacy determination of whether or not this regularization would actually have decreased stroke risk. Is that the primary outcome that you want to look at?
Yes, most like all our patients were under anesthesia for the ablation procedure. So we see that we can make the RR intervals more regular, but we still have to prove that this actually translates to symptom relief. So what we are planning to do, like somewhere once this part of the study completes, we have lots of patients in our pacemaker with persistent atrial fibrillation. And if we design or think about designing a prospective study, they can just come into the clinic. We can adjust the pacemaker settings. They can do either a 6-minute walk test, walk around, they can tell about their symptoms. We can iterate the different settings for the same patient, so we can have a self-control study for this. So probably that will be the next step. Once we finish with 25 patients and we establish that is a safe procedure, then probably that will be the next step.
And in terms of ejection fractions, were you actually able to look at ejection fraction? Were there any changes or benefits in EF?
So
pacing?
yeah, not in this study. Like we use a non-invasive peripheral monitor. This is really just measuring the it can measure the pulse amplitude, heart rate, cardiac output. We don't have in our ablation setup, we don't have a way to continuously monitor ejection fraction. We don't study that.
Okay. Thank you all. Thank you, Dr. Roka, for being brave enough to be first.
Thank you very much.
It was
Okay.
wonderful. It was really wonderful. Thank
Yeah,
you,
it
Dr.
was
Roka.
very nice work. If you want to stop sharing, and then Dr. North is up with his also cardiology related but basic science research.
Okay, everybody can see my slides? Okay, great. So thank you, Dr. Hansen, for the introduction, for setting up this series. I think this is a great idea to help bridge the basic science and clinical faculty within the university. So I'm going to talk about one of the projects going on in my lab, looking at the regulation of intracellular communication by this gene that we study called BUBR1, and a role for it in controlling cardiac electrophysiology. So my lab focuses largely on aging. We try to understand what happens as we age at the molecular and cellular level to try and make some connections with how those changes might lead to whether we get a disease or how severe that disease might be. So shown here are a number of different diseases, many of which are mortality driving, in which you can really see their incidence dramatically increase with age, suggesting that if you can sort of modulate the aging process, you might be able to target a number of these diseases, either individually or collectively. Specifically within the heart, there's a number of changes that occur. Due to time, we won't go through these in great detail. There's significant changes at the functional and structural level, as well as the cellular and molecular level. However, one of the aspects of this is that how the transcriptome and proteome remodeling at the very basic fundamental levels of the cell are changing with age in the heart, and how that might regulate cardiac deterioration. And so the idea would be defining these mechanisms could really help provide novel therapeutic targets for future development. So as I mentioned, we study this gene called BUB-R1. BUB-R1 is traditionally known as a cell cycle regulator. Its primary function is to control the transition from metaphase to anaphase. It essentially prevents the cell from making that transition until all the chromosomes are properly aligned at the metaphase plate. And then it gets deactivated and mitosis can commence. So you can definitely clearly see the connection with its function with diseases such as cancer, where if you lack BUB-R1 or have mutated BUB-R1, you could get chromosome missegregation and aneuploidy. Aneuploidy is also tied to a large number of diseases as well. So if you make a knockout animal of BUB-R1, you get embryonic lethality. These animals don't make it very far into in utero development. If you make a heterozygous animal of these of this gene, you get sort of very few phenotypes that are really overt. It hasn't been studied really in depth. These animals appear to develop quite normally. They will get some tumorigenesis as they age, though. So in an effort to try and understand more about this protein, Jan van Dersen's lab about 20 years ago, he's up at the Mayo Clinic, he made a hypomorphic animal. And for practical purposes here, hypomorphic animals mean that these animals express about 10% of the protein. So they're not completely knockout, but they have very low levels of expression. So this surprisingly is enough to get these animals through development. However, they're stunted in growth, so they're always about half the size of the littermate controls. Then we live to be about a year of age. And they have this sort of premature aging phenotype. You see this kyphosis of their spine. They get loss of subcutaneous fat. They get high incidence of cataract formation. They have difficulty with healing wounds as well. So another interesting aspect that was observed with this gene is that if you look at its expression through the lifespan, you find that it naturally declines with age. And this is, again, coming from Darren Baker's work in Jan van Dersen's lab, where they saw this decline in BUB-R1 with age. And this, over the years, has been documented in nearly every tissue that's been assessed, that BUB-R1 levels decline with age, and it's sort of a natural progress. Those animals I showed you earlier, at 4 months of age, their levels of BUB-R1 are present at about the level of a really old, a 32-month-old animal. So when I was a postdoc and transitioning into my work here at Creighton, I have a real strong interest in trying to understand how does BUB-R1 decline with age. So the idea is that it declines with age. At some point, it's going to reach a critical level where if it falls below that, you get dysregulation of tissue maintenance. This level is going to probably vary depending on which tissue you're actually looking at. But the idea is trying to understand how this process happens, and can we actually delay it or reverse it to prevent the development of age-related diseases. So to make a long story short, and not to get into the weeds too much here, we identified a pathway where BUB-R1 is regulated by a posterior undulation modification called acetylation, and that acetylation promotes its ubiquitination, subsequent degradation and loss. And we believe that this pathway being hyperactivated as you age is one of the reasons why we get this decline in BUB-R1 with age. This process can be reversed by an enzymatic activity of a protein called SIRT2. It's a deacetylase enzyme, so it removes this modification and can maintain BUB-R1 in a more preserved manner. So to test this and whether or not this could really influence these BUB-R1 hypomorphic animals, remember they do express a little bit of protein, so it's actually a good model for this test, is to generate BUB-R1 hypomorphic animals that either lack overexpression of SIRT2 or overexpress SIRT2. Remember that SIRT2, if overexpressed, might be able to remove this acetylation and preserve BUB-R1. And what we found was a doubling of the lifespan of these animals. Shown here, the average lifespan was almost doubled, and then you see a significant increase in the maximal lifespan. If we look at a couple of tissues shown on the right, you see that there is BUBA1 preservation in these animals. These are hypomorphic animals with very low levels, if sometimes undetectable. And then if you increase SIRT2 in those animals, you can preserve BUBA1 and get some benefit to the lifespan. Now, this is a transcriptional downregulation as the mechanism for this hypomorphic alleles. And so these animals are actually producing low levels of protein, so you can only really stabilize what's produced. And so we never really get to wild type levels. So that's why we don't have a wild type level lifespan. So one of the interesting aspects, kind of a tangent that developed off of this initial lifespan study, was that these animals, while all being small in stature, all having sort of these age-related phenotypes, there was a wide range of variability within this cohort of animals. So these are two age-matched animals, and you can see this one has a much more severe kyphosis compared to this one. This one also has more cataract formation, probably more subcutaneous fat loss. And so we saw this wide variation. But what was interesting is that you couldn't really predict when that animal was going to die during the lifespan based on the phenotypic appearance of them. And oftentimes it was the most decrepit looking animals that would actually live the longest in our lifespan. And also you didn't see a deterioration in their behavior, or their physiology, or their health prior to them dying. They essentially just sort of randomly dropped dead. So this suggested to us the possibility that they could be having some heart issues and having some sort of sudden cardiac death. So when we put these animals on echocardiography, we didn't really see any change in the heart function itself. The heart was beating as it should, and comparisons of ejection fractions and those sort of variables were quite similar with these two different, with wild type versus hypomorphic animals. Put on electrocardiography to measure the electrical conduction impulse through the heart, we saw some severe issues with these animals. We found that they have a shortened PR duration, a prolonged QRS, and a prolonged QTC duration. They also had this sort of suppression of this J-point amplitude. And this is sort of on the ECG, the end of depolarization and sort of the beginning of repolarization. This is really difficult to ascertain in mice, though. And so this is sort of a strange sort of part of the EKG, because mice, their heart rate is 10 times faster than a human. So their average heart rate is about 600 beats per minute. So their ventricles are sort of starting to repolarize prior to being completely depolarized from the cycle prior. I put this on here because one of the interesting aspects is when we looked at the SIRT2 transgenic animals, when crossed onto these hypomorphic animals, it was actually this part of the ECG that actually was completely restored. And so it suggests that there might be some impact of this on this process further. However, I'm going to focus mostly on this long QTC phenotype of these animals, because long QTC is really one of the strongest predictive factors for sudden cardiac death in the human population. I'll highlight that this work was done with collaboration by Tony Rosenzweig's lab out at BNDMC at Harvard Medical School. And a lot of this work was done with Michael Rosenberg, who is an electrophysiologist. So we're really interested in how is this happening at the molecular and cellular level, as well as in the animal. So I'll start with first and talking about some of the cellular and molecular changes that we're looking at, and then come to some of the animal studies. So we're really interested in intracellular communication, this process. So intracellular communication can happen at multiple mechanisms, both contact dependent and soluble factors. So contact dependent, you have membrane bound receptors that might interact. You have gap junctions allowing free passage between two different cells, and some other versions of this process. Soluble factors, you obviously have the autocrine, paracrine, endocrine situations, as well as vesicle transport. So we're going to focus on the gap junction aspect of cellular communication. So gap junctions are formed by these proteins called connexins. In particular, connexin 43 is a really predominant connexin within the heart, especially the ventricles. It's a protein that has 4 transmembrane spanning regions. Both N and C termini tend to sit in the inside of the cell. 6 of these can come together to form a pore, or what's called a hemichannel. This pore can then align between 2 neighboring cells and to allow free transport of small molecules between neighboring cells. Within the heart, as far as electrical impulse propagation, it appears that connexin proteins, and as well as connexin 43, seem to play an important role in this electrical signal propagation throughout between cardiomyocytes. Now, when we think about electrophysiology, we oftentimes think about calcium and potassium and channels that are important for that process, but it appears that gap junctions also play a really important role in the electrical impulse through the heart and the conduction system. So these pores can be opened and closed, and a lot of the functionality of these pores relies on post-translational modifications that can happen to these connexin proteins. These situations are observed, or changes in these modifications can happen during various physiological situations within the heart, such as ischemia. So shown on the right is, again, this sort of structural appearance of the connexin 43 protein with the transmembrane regions. You see that this long intracellular portion of the protein, the C-terminus, is targeted by a number of post-translational modifications. And there's a number of different pathways that can modulate those. And in particular, we've been interested in this modification at serine 368, phosphorylation modification. And this has been shown in a number of studies to be really important for the life cycle of a connexin protein, how it gets to the membrane and taken off of the membrane, as well as how well it functions when it's in the membrane in terms of allowing for passage of factors through the channel. So most of the work I'm talking about now is done by a talented grad student in my lab, Ren Poon. She's been working really diligently on this project, and I'm only going to be able to really talk about a small fraction of the work that she's done, but I'll try to highlight some of the other aspects she's been doing. So to test this role of BARBAR1 and connexin 43 phosphorylation in the heart, we first started out with some cardiomyocyte cell lines. This is AC16 cells. They're derived from cardiomyocytes. There's tissue cells in culture, so they don't have the sort of tissue architecture, but we can still sort of study the consequences of these pathways in them. And so she knocked down BARBAR1. You see the reduced levels of BARBAR1. So this mimics sort of the hypomorphic animals. And when she treats these cells with PMA, which is an agent that will sort of increase the activity of a lot of these different pathways that are known to regulate connexin phosphorylation, we see the strong connexin 43 increase in phosphorylation in wild type cells. However, in these cells that are lacking BARBAR1 or have reduced levels of BARBAR1, the ability of those of connexin 43 to be phosphorylated is dramatically reduced. She then wanted to see, well, what's the consequence of this in the cells in the gap junction functionality. And to do that, she utilized a scrape dye loading transfer assay in which you grow your cells in a monolayer in the dish, and you make a slice or a scrape across the monolayer. And then you add a dye called Lucifer yellow to these cells. That dye can enter into the cells through that scrape, but then it can transfer to and migrate through the bilayer or through the monolayer of cells. And you can then determine whether or not you get intracellular communication. And what's unique about this dye is it can only transfer between different neighboring cells through gap junctions. So you can really show exactly how well the gap junctions are working. And so when she did this, she found that the BARBAR1 hypomorphic situation, that there was a increase in dye diffusion rate. So the cells that lack BARBAR1 that have this reduced level of connexin 43 phosphorylation, that those gap junctions are working a little bit better than before. So they're allowing more transfer of dye through them. So future directions that she's working on in this particular area at the molecular and cellular level is really trying to understand how BARBAR1's regulating this process. And as I mentioned, we can't really go through a lot of her work, but she's started to really pin down a novel kinase that hasn't been involved or shown to be involved in this process that BARBAR1 might be targeting to regulate connexin 43 phosphorylation. So to get into some of the in vivo work that we're interested in doing, just showing this slide to remind you that these animals are hypomorphic animals. They have a loss of BUBR1 in all the tissues in the body, right? So one of the problems with studying these animals is we really don't know whether or not defects we see in an organ like the heart, if that's due to an intrinsic function of BUBR1 in the heart, or if it's through some function that it's having in another tissue that's impacting the heart. So to get around that idea, we've generated a conditional knockout mouse model where we've flanked exon 4 with LOXP sites. And then once you cross those animals to another animal that expresses CRE in either tissue specific or a time dependent manner, you can target BUBR1 and knock it out in a tissue specific situation. So to do that with the heart, we utilize this gene called NKX 2.5. This is a gene that's almost exclusively expressed in the heart. There are some tissues where there is some very minor expression, but largely it's a early transcription factor involved in heart development. So it turns on about E7.5 in mice, which is right around the time that the heart develops, right? And shown here is this mouse model that we're using, this NKX 2.5 CRE, was developed in early 2000s. And you can see here that there's, using this reporter mouse, that its expression is really fixed in the heart. So Ren has tried to identify what happens when we knock out BUBR1 in the heart, right? What's the consequence of that? So what she did is she took BUBR1 hypomorphic animals, one of which would express homozygous NKX 2.5 CRE. When she crossed those up together, we anticipated the traditional Mendelian ratios of 1 to 2 to 1 of the expected BUBR1 genotypes. However, what ended up happening is that she never found a wild type complete knockout or homozygous knockout of BUBR1 in any of her crosses. So shown here are 6 different crosses she did with this particular pairing. She's done a number of different switching how the pairings were done to see if there's some variability in that aspect, but we've never gotten a viable animal that has BUBR1 knocked out specifically in the heart, which really suggests that BUBR1's role is really critical in regulating heart development. But we have wanted to move on and see whether or not we could still use the heterozygous animals, right? So haploinsufficient animals, do they still have the consequences that the hypomorphics have? And so what we did here was to use heterozygous animals that are either lacking the NKX 2.5 CRE or having the NKX 2.5 CRE. And what you see here in the heart, you see about half the expression of the protein, or maybe a little bit less. Whereas in other tissues such as the liver, you don't see that change in the protein levels. And when she did surface EKGs on these animals, she did find that they also have a prolonged QTC duration. So it suggests that there might be some value in these animals, and we can sort of start to assess these animals while we still develop newer technologies to really try to knock it out still completely in the heart. And so what we're in the process of doing, and this is really kind of highlighting some nice collaborations between the clinical side as well, as we've been working with Dr. Michael Kim at trying to understand how we can monitor what's happening in these animals. So as I mentioned, these animals are dying at about, the hypomorphic animals die fairly shortly, but the real issue at hand is, are they really dying of an adverse cardiac event, or are they dying of something else? So to try and catch that, we've been collaborating with Medtronic. They have these linked devices that are typically implanted subcutaneously above the heart in humans to long-term monitoring. That we've been able to get some of those that have been repurposed, and shown here is Ren doing a survival surgery of these animals, and here's an animal that now has this linked device implanted on the back of the heart, and we're able to get good readings of the heart function. So we're now in the process just waiting for these animals to die, and then to hopefully see when they do die, what is the sort of functional consequence of that. So some future directions are to continue on with these animals, try to understand what's really going on in them. As I mentioned, there's a lot of weird variations in their conduction. So like they have a prolonged QTC, but their PR duration is shorter. So, there seems to be sort of a differential impact that BUBR1 might be having in each of the different chambers of the heart. So trying to understand these will really benefit from having sort of a clinical perspective, or a more perspective for what might be seen in humans that might mimic some of the aspects that we see in these animals. So with that, I'll just quickly just acknowledge some of the people doing this work. So I have a great lab of people that are all really collegial and have contributed to this project as well. Most of the experiments I showed were done by Ren, as I mentioned. We have some great collaborations already with the School of Medicine. We'd love to have more. We have some great collaborations with Medtronic as well. So with that, I'm happy to take any questions anybody might have.
Thank you, Dr. North. Really nice work. You have any preliminary information about the heart rhythms in those animals?
So we're just waiting for those animals to die. You know, the real issue here with this device is this is really the first sort of implants is really a test. Like, is it going to work? Are we going to be able to detect anything? But these devices are really developed for humans, right? Who have a resting heart rate under 100, or well under 100, right? These animals have a 600 heart rate. So there's been some opportunity to try with Medtronic to try and reprogram them and how they're, you know, what their sensing cutoffs are. But that's really, it's really kind of a challenge to know, you know, where that cutoff should be. So it's not over sensing or under sensing. Under sensing what's going on in the heart. So that's kind of where we're standing right now. We do have them in. We're waiting to see how that process is kind of working out. Just taking time to get uploads and to analyze how the sensing is going. But apparently, from Grant's perspective, this is the smallest animal that they've implanted these into. He talked about implanting them into a bear at one point. So, you know, this is much easier, I think, than his bear implants. But
Dr. North, I think you had a great presentation, and I had a question more so not on the content, but your career development. I understand that you have a KO1, and just was curious if you'd be able to speak to just a little bit about your career development and how you transitioned to that and made a successful environment to be awarded a KO1 here at Creighton.
Yeah, so I received the KO1 back when I was at Beth Israel, and I was able to transfer it to here. So the KO1s are kind of interesting. I think, you know, you always have to have great mentors, right? That's really a key to any career development grant. I think it's key to any grant you have in any point in your career. As an aside, like, as I always like to say, when Dr. Hansen's on the call, I actually worked with her when I was a undergrad, and you know, it's great to have great mentors like that all throughout your career. KO1s are, you know, they're really kind of about a third about the project, third about you and your career development plan and past experience, but a lot more about the plan you have in place, and then a third about your mentor and how that mentor is really going to help promote you and promote your work and your effort, right? So I think that's kind of the key about the KO1 awards. But they're, you know, K awards are really really important for the transitions between your postdoc or early career, you know, maybe non-tenure track faculty into a tenure track position.
That might be a good discussion to take offline too, or, you know, into the mock study section reviews. It seems like you have a lot of opportunity for clinicians or students or residents, fellows, who might be interested in getting more involved.
Yeah, I think it would be fantastic. As I mentioned, when I was at BIDMC and doing these studies, we collaborated with Tony Rosenzweig's lab. He's a really great person in the cardiac field. And Michael Rosenberg was a fellow at the time that he was helping us with these. So, you know, be fantastic to get that expertise. You know, he was able to look at these ECGs and kind of really give us a lot of insight into what's going on, because we just don't have that expertise, right? To just even just viewing them and trying to understand what the data means, right? And making sure that we're viewing them properly and analyzing them properly. That's something that I think it takes electrophysiologists, or, you know, years to master, right? And we're trying to do it here, but also trying to do it on an animal that has 10 times heart rate, and the ECGs aren't as defined as they are necessarily in a human ECG.
You know, Brian, this is Maureen Tierney, Chair of Clinical Research. Great work. And in terms of overall sort of you bringing your K01 here, but still yet having a connection, I actually trained in ID at MGH at the same time Tony was doing his fellowship there. So I know Tony Rosenzweig really well. And, being able to potentially have him, Laura, as a, an official sort of external mentor or trying to, develop even further collaboration, I think would be a fantastic opportunity.
Yeah, and I think going back to all these kind of talks about career development is like mentors. Another sort of interesting thing that happened in this progress is we kind of stumbled into this whole field. And so I have great collaborators like Tony, who are really at the clinical side, but I also, when I was a graduate student, I did my first rotation in a lab that I had no intention of joining, but it was a person who studied heart development. And it just turns out now we're in a situation 20 years later of looking at heart development. We've reached out to him, he's at UCSF, and we've had some great conversations with him about these projects. And you never know when you're going to have met someone in the past who's going to be really beneficial and a good colleague in the future, right? So I think that really speaks to maintaining good relationships and having good mentors all throughout your career, and not pigeonholing yourself into a certain area of basic science, but being open to seeing and following where that data is taking you, right? We have this mouse model that has premature aging phenotypes, and we're interested in how these sirtuins are regulating them, but lo and behold, there's this whole new area of what is this gene doing in the heart. And by Ren's work of knocking it out specifically in the heart, it really shows a real important role for it in the heart development. And trying to understand that is going just well beyond where I've been in the past. So it's always a learning process.
And I gotta say too, since there are means to increase CERT activity, that might be a clinically relevant treatment for some populations. That would be really interesting to talk about too. So
yeah,
we should have
it'd be
a
interesting
cardiology research discussion.
Right. I think it'd be interesting in the context of this J point, right? So when we did this study, the J point was just sort of this obscure part of the ECG. And I think it still is kind of a really obscure part of it, but there's enough information out there that might sort of really pinpoint, and maybe this studying SIRT2 and the role of BAR1 might help us to understand that impact, because you can extend these animals' life. And that was really the only attribute of these ECGs that was really changed in that context of that study. So be interesting to see if how that really plays out.
Might be interesting for future stuff. Okay, it really great talks. Thank you both, and thank you everybody for joining us today. We're past our allotted time, so please send me any suggestions for future talks. And thanks so much for going.
Laura, can I just add one quick thing? Sure. Dr. North, just want to sort of say that what you said about being open to different areas of research, Sandy Byers is the major contact to be connecting students with mentors. And she often encourages students, if there's not something right in their area of interest, to be very open to the research process in general. And so your story is actually underscores that advice. So thank you for sharing that.